Hi, how's it going, guys? Our last video for the school year, we're going to talk about organic chemistry, which is really going to be the bridge from chemistry this year to biology, which you are going to take next year. 7 learning objectives that we're going to look at. We're going to focus mainly on these middle learning objectives in the notes. When you do your POGIL activity in class, we'll look at 15.2, and then next week we will work on 15.7. So let's take a look at just some basic information about organic. So the basics of organic molecules, they're kind of 2 ideas. And in chemistry, most of the year, we've been looking at inorganic or non-living molecules, so water, oxygen, things like that. Biotic or organic means living or once was alive. So those things we often find and know contain carbon. And so organic compounds are covalently bound compounds containing that carbon, which is essential kind of to life. Characteristics of organic molecules, they have low melting points. Think of sugar, it melts pretty easily, as opposed to salt. You can't melt salt on your stove. Highly combustible and flammable. Again, we use oil and things in our gas or in our cars, which were from fossils from the dinosaurs way back when. And they're also insoluble in water soluble, but they are water soluble in benzene and alcohol, which we'll talk more about next year. So when we talk about the basics of life, all organic material is made up of these 6 molecules, and we can abbreviate or think of them as SHNOPs. So carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur are the main components of life. So everything that's making up your body right now wildly is mainly these 6 elements. So what we're going to do today is take a look at the differences between what the monomers, meaning the simple small molecules, and the polymers are for the 4 major macromolecules. So again, monomers being those base units, and the polymer being a combination of those base units. The 4 macromolecules that we're going to look at are carbohydrates, lipids, proteins. The 4 macromolecules are carbohydrates, lipids, proteins, and nucleic acids. And we're going to talk about carbohydrates first. So carbohydrates are very complex and they make up a lot of life. You think about them often when thinking about eating things. And the monomers of carbohydrates are simple sugars. It's the subunit of those carbohydrates. And the basic one we often hear about is glucose. Carbohydrates come in a ratio of 1:1 of carbon, hydrogen, and oxygen. So carbohydrates are made up of those 3 atoms in various ratios with each other, but it's always a 1:2:1 ratio. So if you look at ribose here, we have C5H10O5, which if you simplify that down, it's a 1:2:1 ratio. So that is our kind of empirical formula, if you remember back to that, the molecular formula then being that ribose. Glucose then C6H12O6. Disaccharides, which we maybe don't know of as disaccharides but do consume often, are when we have 2 monosaccharides or 2 monomers of sugar connected together. And they connect together through what's called a condensation reaction, and we are going to look at that in our POGIL this week. So when we have 2 glucoses coming together, we form maltose, glucose, and galactose, which is another monomer of sugar. You form lactose, and this is milk, guys. So if you're lactose intolerant, it's because your body can't process lactose into galactose and glucose, which your body then can use as energy. So your body struggles with breaking down this sugar into these 2 sugars. Glucose and fructose form sucrose, which is our table sugar that we know of. And so here's just a picture of that condensation reaction. We have 2 glucose molecules coming together here to form a maltose here. So another little diagram here. We often consider carbohydrates in 2 categories, simple and complex. And the 2 different simples are those monosaccharides, which are the monomers, and then disaccharides, which is sort of a polymer. It's a basic polymer. Glucose, fructose, and galactose are the monomers of carbohydrates. They can combine then, like we just looked at, into maltose, lactose, and sucrose. The complex carbohydrates are known as polysaccharides. Poly means many. So many of these monomers together can form starches, fibers, and glycogen. Glycogen is how we store complex carbohydrates in our body. Starches and fibers are how plants store complex fiber. So those potatoes you're eating that is very starchy have a lot of energy in them because there's a lot of monosaccharides, a lot of glucose that have been combined together to form that starch molecule. So, can you diagram the structure of basic carbohydrates? What are they made of? They're made up of carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. Let's move on to lipids, our second macromolecule we're going to talk about today. Structure and function. Lipids are found in every type of plant and animal because they make up the cell membrane. You've probably talked about that before. They contain, again, carbon, hydrogen, and oxygen, but they have much less oxygen than carbohydrates, which actually leads to them being insoluble in water. They often don't mix. It's kind of like the oil and water example here. Like I said, they're in the cell membrane. Their hormones, their energy, storage and molecules, thermal insulation, mechanical protection, electrical insulation of the nerves. Your brain is mostly made up of fat, and waterproofing and buoyancy. So a lot of animals will use this, like ducks, to stay warm in cold water. So there are kind of two different types of fat structures we're going to look at. We're going to look at the difference between saturated and unsaturated fats. And so when we talk about this, usually you've heard this in terms of health and being healthy and whatnot. Saturated fats are fats that do not have any double bonds in them. And saturated fats are going to come from animal products. Saturated fats are going to be solids at room temperature, and that's because they can neatly pack close to each other because of those. There's no presence of a double bond. Unsaturated fats, or trans or cis unsaturated fats, which is going to change the shape of that fat, is when there's a double bond present, or multiple double bonds in the fat structure, which is going to change the shape. So instead of being a nice straight line, which is going to pack together, unsaturated fats are going to bend and work around each other. These are going to be your liquids at room temperature. We use, like we said, in our cell membrane, what's called a phospholipid bilayer. So the phospho part comes from this phosphate and glycerol head part, and the lipid bilayer part comes from these two lipid tails here, which are made from a fatty acid here. And so this one you can see is saturated. There's no double bond. It's nice and straight. This one is unsaturated, has a nice little bond here. You'll talk a lot more about the cell membrane in your biology class, but this part of that lipid is going to be hydrophobic. It doesn't like water, just like we talked about. Lipids do not dissolve in water. So, can you describe the differences between the structure of saturated fatty acids and unsaturated fatty acids? Let's move on to proteins. So proteins are made up of a monomer of amino acids. Amino acids are really important to the building of our bodies. Our bodies are made up of a significant amount of protein, and they are made up of various amino acids. Amino acids have 4 parts. They have this amino group, which has this nitrogen present. They have a hydrogen group, and then they have a carboxyl group on this end. Then they have this thing called an R group. So all amino acids have the blue, purple, and green. The yellow varies from each amino acid to each amino acid, which is what gives it its characteristics. So like tryptophan, glycine, those are 2 examples of amino acids. When amino acids bond to form a polymer, they do so with a what's called a peptide bond. So a bond forms between the oxygen, hydrogen, and hydrogen here. A water molecule is released, and we formed this blue line here, which is the peptide bond between these 2 amino acids. So in this picture here, we have a red or a pink, green, and blue amino acid being bound together with a peptide bond to form a strand of protein here. Those strands of proteins then get wound and weaved together to form a very complex protein. Proteins are really complex. They have 4 different kinds of structures, and they make up most of life. So when we talk about amino acids, there's some information out there about essential and non-essential amino acids when talking about kind of your diet. The difference between them being, we call them essential because the body cannot make them. So there are amino acids that our body cannot make and you must consume in your diet. And this becomes really important if you are vegetarian or vegan, because many of these amino acids are really easy to find in meat products, but very difficult and are in low quantities in vegetable products. So again, they must be obtained through diet. They're available in animal products. So like I said, you have a really hard time getting your essential amino acids in the amount you need. If you are a vegetarian or vegan, you have to be very conscious of this. And these are the examples of the essential amino acids here. And then non-essential ones are that your body can actually synthesize and make in order to produce more proteins. And there's quite a few of those examples there. Protein structure, like I said, gets really, really complex. So there's primary structure, which is your polypeptide chain, which is made up of those amino acids being bound together. Secondary structures, then that chain goes through a change where it either is forming an alpha helix or a beta sheet, and those can form some different things as well. The secondary structure then gets bound or connected to some other structures with different bonds, so that alpha helix and that beta sheet get connected to each other and shape and form into different shapes. And quaternary is where you're then forming multiple bonds with multiple strands of protein. Hemoglobin is a really good example of this. It looks like a donut, and that's your red blood cell. And the whole purpose of that donut is the middle part, that hole, it's not really a hole, it's a divot, but is to capture oxygen. That hemoglobin is what carries oxygen through the bloodstream. So that whole protein structure's job is to attract and attach high oxygen to it, and then be able to detach that oxygen and give it to the living cells in your body. So, can you explain how amino acids can link together with peptide bonds to form proteins, and what are those proteins and what do they look like? So last but not least, we have nucleic acids. So there's 3 parts in a nucleic acid. We have the phosphate group, we have the pentose sugar, and we have the nitrogenous base. Notice all these nitrogens, okay? So those are those 3 parts of our nucleotide. Nucleotides are our DNA and RNA. They are the information that makes you you. So DNA, which if you haven't talked about this much, or you will in bio, is double-stranded. It's stands for deoxyribonucleic acid, because the ribo is the sugar and then that nucleic acid. RNA is ribonucleic acid, so the structure is just slightly different and it's single-stranded. The amino acids in DNA are cytosine, guanine, adenine, and thymine. The the nucleotides in RNA are the same, except for instead of thymine we have uracil. So that's kind of the main differences between those two nucleic acids in those 2 molecules that are essential for life. The monomers then of each of these guys, those monosaccharides, so those basic sugars like glucose, then the polymers would be a complex carbohydrate like starch, or even lactose in milk. Fatty acids then are going to be your single chain here. A lipid then is made up of combinations of fatty acids, whether they are unsaturated or saturated. Nucleotides then are those base units. We have thymine, adenine, guanine, uracil, and nucleic acids then are made up of a combination, so that DNA and RNA that make up in the nucleus of our cells. Amino acids then are the monomer or building block for proteins. Proteins then are combined amino acids in a peptide bond and then form very complex structures that make up most of life. So can you explain the composition and function of a nucleotide? Again, looking at all 4 of our macromolecules. Finally, this is our list of objectives we started with. So we kind of looked at 15.3, 4, 5, and 6 specifically. 15.7, constructing and revising an explanation based on evidence to explain how the structure of carbon-based molecules impact their function. We're going to work on this next week. We're going to work on this in class and try to develop some information on these, okay? Awesome, guys. Last video, we made it. Last learning target of the year, really bridging you between chemistry to biology and how they are deeply connected with each other. It's been great getting to know you guys, and it's kind of sad the year is over already, but it will be good to have a nice summer break between us. So work on finishing up the notes for this. I'll see you in class. The only assignment for you guys asynchronously are these notes this week. The other 2 assignments will be done in person in class, which is the POGL, and then the kind of poster assignment that we work on 15.7 and 4. As always, if you have questions, comments, and concerns, reach out via email.